Today we're going to be talking about non-Mendelian genetics. So we'll be able to contrast these genetics relationships with Mendelian genetics. And remember that Mendelian genetics is complete dominance, and that's where you have one allele that completely overrides the other. So your dominant allele completely masks the recessive allele. So the examples that are given in your Bio 101 textbook are some of these. And again, just to keep in mind what we talked about in the last lecture is that for these you actually have a gradation of phenotype. You don't either have freckles or not have freckles. You can have lots of freckles, you can have a little bit of freckles, you can have completely attached ear lobes or gradations of attachment and detachment. Widow's peak can be really sharp, can be kind of barely there. So just to keep in mind as you're reading through the book that these really aren't complete dominance where you either have it or you don't. So let's talk about, how you'd have something like gradations of widow peak. We're going to be talking about different genetics relationships. We're going to be talking about incomplete dominance. We're going to talk sex-linked genes, polygenetic inheritance, epistasis, as well as codominance. So to begin, let's start with incomplete dominance. And our example that we're going to do is hair curliness. So first we'll look at Caucasians. First, let's look at our nomenclature. So your homozygous dominant genotype will produce curly hair. Your heterozygote will give wavy hair, and your homozygous recessive straight. So notice the difference between incomplete dominance and complete dominance. If this was complete dominance, then this genotype would produce curly hair. Incomplete dominance occurs when one allele, the dominant allele, its presence doesn't produce the full phenotypic effect. So you only get part of the phenotypic effect. You get wavy. Now in Caucasians, what you have is this whole spectrum of curly to wavy to straight, right? And so you have allele variability. You've got big H's and little h's in the Caucasian population. Now, for hair curliness, what that means is that each individual follicle, if it's straight, it's round. And the flatter it is, the more curled it is. So a really flat would be like running a ribbon over scissors. As the hair grows out, it curls. And then you can see down here that follicle size for hair thickness, the differences there. So what about other descents? Let's look at African and Asian hair. For African hair, you have universally collapsed follicles, and so there isn't the allele variability that you find in Caucasians where you have, round open follicles, partially flat follicles, and completely flat. Here you just have completely flat, and so as the hair grows, it curls. Now for Asian hair, what you have is universally really open, wide follicles. So Asian hair tends to be thicker, each individual strand, and then very, very straight. And again, that's because there isn't a diversity in the alleles that code for hair curliness. Let me show you this in practice. You'll be doing problems in class where you will have to figure out incomplete dominance ratios. So you're going to draw Punnett squares for these just like you did for complete dominance. The only difference is that you're going to read it different. So if we're looking at something like flower color and we've got red is dominant to white, okay? So I've got a red and a white allele, I've got a heterozygote. And then let's have that heterozygote mate with another heterozygote. We go ahead and we fill out this table. And what you can see here is the genotypic ratio is just like it would be for complete dominance, right? It's 1 to 2 to 1. So 1 to 2 to 1, right? but the phenotypic ratio varies. If this was complete dominance, what you would have is a 3-to-1 red to white. But if this is incomplete dominance, you read it different. It's 1 red, 2 pink, 1 white. So let's get that down. You've got red and you've got intermediate now, pink and white, 1 to 2 to 1. Okay, so that's how you solve those problems. All right, so that's incomplete dominance. Next, what we're going to look at is sex-linked genes. All right, so this is figure 1111 in your book. And for sex-linked genes, basically what we're going to be dealing with are the X and the Y chromosome. So remember that if you're female, you're XX. If you're male, you're XY. And the issue here is that when you have traits that fall on the X or the Y chromosome, there's a unique situation that occurs, not necessarily for the female, but for the male. So notice that males, because they're XY, there is not a homologous chromosome, a homologous pair. Females, yes, you have a homologous pair, XX, right? So alleles can duke it out. But the alleles, there aren't alleles. The genes don't line up. This is a totally different chromosome. So for males, there's kind of a special situation when you're figuring out these problems. So here you can see a Punnett square that's done, and we're going to do pedigrees instead, and I'll show you that in just a minute. But what you can see here is, you know, a female can be healthy. A female can be, a carrier. And a female, which isn't shown here, can actually be an affected. But the odds of the female being affected are less because you have at least one healthy allele. You can actually be a carrier. For males, you can't be a carrier, right? This is not a carrier because there's no homologous chromosome. So there's nothing to duke it out. If you have it, you have it. And so these X-linked genes that fall on the X chromosome are much more likely to show up in males. It doesn't mean they can't show up in females, but it's much less likely. It's much more likely to occur in males because you don't get the carrier option. There's only two options. You either don't have it or you have it. Whereas females, either you have it, you're a carrier and healthy, or you don't have it and are healthy. Okay, for this example, I'm going to show you male pattern baldness in my family. So to do this, I want you guys to draw out pedigrees, and I'll make a caveat here that I really don't want you to use shading, and that is because of the nature of sex-linked. You're welcome to use shading with any other pedigrees that we do, but when you're dealing with the X and the Y chromosome, it's best to go ahead and write it out. Okay, so what we've got here is, this is my family. This is a circle, so that means that's my mother, so I write XX in there. And then this is my father, which is a square, and he's XY. And what I know is that my mother's father was a carrier for male pattern baldness, so I needed to put a little B there. I'll show you why that is in just a minute. And my father had male pattern baldness, so I put his little B there. And male pattern baldness is on the X chromosome. And so they had me, and I'm female, so I'm XX. And so this is where pedigrees are particularly helpful. What you can do is you can look and see, okay, well, if this is dad right here, then I'm female, so he had to give me his X. He didn't give me his Y. So he gave me that X, and that automatically makes me a carrier. So females, if your father has male pattern baldness, you're automatically a carrier because you're female and he had to give you the X, his X chromosome. If you were male, he would have given you his Y. From my mom, I don't know what I got. So this, she could have given me this X with the little B on it, but I'm just going to go ahead and choose this one because it doesn't have a B on it, and that sounds better to me. So females can get male pattern baldness. They just have to have 2 of these B alleles. Okay, so I've got myself as a heterozygote. My husband, on the other hand, his mother's father did not have male pattern baldness, and his father does not have male pattern baldness. And he does not have male pattern baldness because his dad gave him a Y to make him male, and his mother gave him one of these clean X's, right? Okay, so we have a son, Grayson, and since he's a boy, his father doesn't determine whether or not he has male pattern baldness because the father gives a Y. Who determines this is the mother for the males. And so mothers, we pass this down to our sons. And so if I'm a heterozygote, the odds of giving him, the odds of him having male pattern baldness, since he can't be a carrier, is 50%, so 50-50 that I gave him this B. And so we won't know that until a little bit later. And that's my son. Okay, let me help you out here in terms of drawing pedigrees, because sometimes students struggle with this. So what I recommend doing is going out to the grandparental stage. So let's say that we're talking about a daughter and we're trying to answer questions about her in terms of whether she's a carrier for male pattern baldness or whether she's going to get it. So you just start with the child and then make a V and put father on one side, put mother on the other side, and then V again. Mother, father, mother, father. Okay, so now you've got grandparents, right? The next thing you would do is go ahead and fill in X's and Y's. So again, you don't want to do any sort of shading. X's and Y's, XX, XX, XY, XY, XX. And then what you would do is just read the criteria in the problem and put in your little B's if you're dealing with male pattern baldness to try and figure out that puzzle. So that's how I suggest setting that up. Okay, next is polygenic inheritance. Polygenic inheritance has to deal with multiple genes coding for a trait. So the phenotype is produced from multiple genes. In this example, this is skin color, but this also pertains to things like height in humans as well as eye color. And what I've got here are 2 intermediate phenotype individuals, and I know that because there's a dominant allele for each gene. So gene 1, gene 2, whoops. So gene 1, gene 2, gene 3, and then gene 1, gene 2, gene 3. So if we say this is mother and we say this is father, they're both heterozygotes for all 3 genes. Now all 3 of these are pigment producing genes. So you've got some pigment production in all of these. Now it's not complete dominance where you either have pigment or don't have pigment. It's cumulative. So the more capital letters you have or the more dominant alleles you have, the more pigment that's laid down. So the darkest possible genotype would be big A, big B, big C, big C. And that would produce very, very dark. And then the lightest would be little a, little b, little c, and that would be very, very light, although not albino. All right, and how you answer these problems is you don't really need to do a Punnett square. You can kind of cherry pick alleles, and I'll show you how to do that. So if I go ahead and write our phenotype on here, our genotype, so let's say we've got heterozygote for the A gene, homozygous dominant for the B gene, and heterozygote for the C gene. And then that is paired with someone who is homozygous recessive, heterozygous, and homozygous recessive. And let's say the question I'm asking is, what is the darkest possible phenotype that these 2 individuals can produce? what you're going to do is basically meiosis for each gene. So you're going to segregate alleles. So you're going to cherry pick though. You're going to choose the ones that would give you the darkest possible phenotype. So you'd go to the A gene and you'd say, okay, I have a big A. So yep, absolutely. Let's give that child the big A. We're trying to produce the darkest possible phenotype. So that's one allele from mom. So that would be packaged in the egg. This would be, one of these would be packaged in sperm, and you only have one option, so you got to transfer a little a down. So there you go. In the zygote, you would have big A from egg, little a from sperm. And then with the B gene, you've got a big B, and you've got an option here. We're making the darkest, so give it the big B gene. So dark, dark, dark. We're up to 3 dark alleles. And then for the C gene, there's a 4th dark allele, but you have to give a light allele for the C gene. So that would be a correct answer in terms of producing the darkest phenotype. Okay, epistasis is our next one. And for epistasis, what you have are 2 genes. So polygenic is 3 or more genes. Epistasis is only 2 genes. And what happens here is you have a gene that codes, in this example, for color. And then you have a regulatory gene that goes ahead and regulates the production of pigment. So this regulatory gene says either, yes, go ahead and lay down pigment, or no, do not lay down pigment. Notice that within both of these genes, it's complete dominance. So, and you know that because the heterozygote is producing black, there's not an intermediate. So for color, for mice or for Labrador retrievers, same thing. You can have black, black, or brown. So you might be thinking to yourself, how do you get white or how do you get a yellow lab? That's where this gene comes into effect. If this is your genotype and you're a heterozygote for the B gene, that says black, right? So that one right there. But it's paired with homozygous recessive, and that says no pigment. So it turns that gene off. The pigment is never even produced, and you end up with white. So you need to combine those 2 genes to understand epistasis. The last one we're going to look at is codominance. And for that, we're going to look at blood type. We're not going to look at Rh factor, just blood type. So that is A, B, AB, and O, right? So first what you need to know is that A and B are dominant to O. And you can see that as evident here. An A-O genotype is type A, B-O is B. But look how interesting this is. A and B are codominant to each other. So when you have an A and a B genotype, A and a B allele, then you get both. Okay, and then the only time you can have type O is when you have this type of genotype. So let's take a look at blood to begin with here. So what we have here is a picture of kind of what constitutes blood. We're going to be worried about red blood cells certainly, but what we're also going to be worried about are antibodies, and antibodies are in the plasma. Let me pull up this image here. All right, so what we want to know about is how genotype relates to phenotype. Here you have group A, and so if you have type A blood, it means that on your red blood cells you have these little A antigens, which are little flags that communicate with your immune cells. If you're type A, you make antibodies, which are immune cells that are type B, to make sure there aren't any B red blood cells in the blood. So one thing you might do is just copy over this and write plasma. So you could write plasma here for this whole row. All right, and then these are red blood cells. Okay, so if you're group B, you have B flags and you have anti-A antibodies, right? And so on and so on. So things that we want to know is how does this relate to things like blood transfusions? When you donate blood, plasma, red blood cells, those are separated out. It increases the number of uses you can have for blood. So if you go ahead and let's say you're group A and you need red blood cells, I'd like for you to be able to figure out which type would be okay for you to get. You have anti-B, so that means that you can receive your own. So if there are type A blood cells, you could get those, but you couldn't get B because these antibodies go after B antigens, anti-B. So if you can make a match, it means you can't use it. Okay, so no. AB, you still can't use it because there's B right there and B, and these would attack those. But you could receive O because the antibodies won't react to anything because there aren't any antigens on type O. Okay, so let's look at plasma now. So that's for red blood cells. So your group A, let's say that you need plasma. Let's say you have an autoimmune disease and you need new blood plasma. Okay, so let's look at this again. Let's see, can you get group B's plasma? In this case, you would just be looking at the plasma row, okay, and comparing it to A, both of the boxes. So you would not be able to receive group B's plasma because it has anti-A and this is A. You would be able to receive AB's plasma because there aren't any antibodies to attack. You would not be able to receive group O's because it has A and B and the A's would match up. Okay, the B's would be fine. Okay, so we're going to work through problems like this for codominance. Okay, that concludes the lecture on non-Mendelian genetics. We'll do more on this in class.